Hello girls, in today's video lecture we are going to learn about specimen observation techniques in light microscopy. So we all know that microorganisms cannot be visualized with our naked eye. When microorganisms cannot be visualized with the naked eye, we use an instrument called as microscope to observe these microorganisms. So can we observe these microorganisms in unstained conditions? No, we cannot visualize them in unstained conditions because these microorganisms, they consist of a clear protoplasmic or cytoplasmic matter. So if we have to study about these microorganisms, that is the shape of these microorganisms, the arrangement of these microorganisms, it is always necessary that we develop a contrast to these microorganisms from their surrounding environment so that we can observe them easily. So what do we do to observe these or visualize these microorganisms? We carry out a technique which is called as staining technique, generally to develop a contrast and observe them under the microscope. So when we talk about mainly preparing specimens in light microscopy, so we can prepare specimens in two basic types. The first important type is called as wet mount and the second one is staining techniques. So what is a wet mount technique? Wet mount technique is a technique where the bacteria or microorganisms are directly observed under a microscope in live conditions. It is the simplest type of preparation in which the specimen is placed on the slide in a drop of liquid. So what we can do here is a broth culture of the bacteria is taken which is present in a liquid form and it is generally added onto a slide and with the help of a dropper, and then it is mainly covered with a cover slip. So this drop of culture is placed between the microscopic slide and the cover slip and it is then visualized under the microscope. What is this technique used for? Mainly this wet mount technique is mainly used for observing the motility of the bacteria. So it mainly helps us to visualize the different types of bacterial motility. So how do we carry out this technique of hanging drop method or wet mount technique? So when we talk about this hanging drop method, which is mainly used for observing bacterial motility, the procedure says like this. We are going to suspend a small drop of the given suspension using a Pasteur pipette to the slide. Then we are going to place petroleum jelly on the side edges of the cover slip and gently press the cover slip onto the drop of culture. Then we are going to place the entire slide on the microscopic stage and observe it under low power. So we are going to adjust the source of illumination, that is mainly through iris diaphragm, so that small amount of light is admitted and then we can observe the microorganisms clearly. But under low power they are just observed as tiny dots. So the objective can be mainly transferred to a high power objective and then the motility can be observed clearly using a high power objective. So this way of unstaining the bacterial organism and trying to observe bacterial motility is called as the hanging drop method, for which is a wet mount technique in observing the microorganisms. Now coming to the next important type of techniques in light microscopy, we have discussed that the second important type of technique is staining techniques. So now what is a stain? A stain is nothing but it is a dye which is generally used to color the living or the dead organelles. So it is mainly used to color the living or the dead microorganisms, and these stains can be categorized into three important categories. Generally they can be acidic stains, they can be basic stains and they can be neutral stains. So acidic stains generally they have a negative charge on them, and examples of these acidic stains are eosin, then acid fuchsin, malachite green, nigrosin, Indian ink. All of them have an acidic charge, that is a negative charge on them. basic stains. So when we talk about basic stains, they are mainly positive, they have a positive charge, and when they have a positive charge, they stain the which part of the cell? They stain the negatively charged components of the cell. So for example, nucleic acid is negatively charged. It is a main important component in a cell. So these basic dyes which are positively charged stain the negative component of the cell. So examples are methylene blue, okay, crystal violet, gentian violet. All these are examples of your basic stains. Neutral, they have both positive and negative charge, and they impart different color to different components. Example, Leishman stain, what we generally use in identifying the blood cells present in blood. Giemsa stain, Wright stain, all these are examples of your neutral stains. So when we talk about basically the first important one, that is mainly stain, which is a colored compound and which gives a color to the microorganism. They can be categorized based on the charges that are present on them. They can be categorized into acidic, basic and neutral stains. So here are some important examples of basic dyes. Once again, methylene blue, okay, basic fustian, crystal violet, safranin, what we mainly use in case of gram staining. All of them have a basic charge, and these basic charge basic dyes, as I told you, they bind to negatively charged molecules such as nucleic acids, many proteins and surfaces of bacterial cell walls. Similarly, the next important one, acidic dyes. So when we talk about acidic dyes, the best examples are eosin, rose bengal, acid fustian, and they mainly bind to positively charged cell structures. Now let us learn about the types of staining methods. So when we talk about the types of staining methods, there are two important types of staining methods. The first one is positive staining. What do you mean by positive staining? Positive staining is mainly where the cells are mainly taking up the stain and they appear colored under a clear background. So here we are trying to stain bacterial cells. The bacterial cells are taking up the stain and they are observed under a clear background, that is a bright background. This is mainly called as positive staining. So when we talk about the various categories of positive staining, we have simple staining again. So where we see simple staining mainly is nothing but it uses only a single dye to stain the organism, and mainly it is used for observing the morphology of the organism. Morphology is the size, the shape and the arrangement of the organism. A single dye is used. So that is why it is mainly called as simple staining. Here we can see that methylene blue, diluted carbol fustian, all of them any form of dye can be generally used for simple staining. When it comes to differential staining is a stain which differentiates between the bacterial groups, or it differentiates between the various structural components within the bacteria. So that is what is mainly called as differential staining. So it can be categorized like this. It will differentiate the bacteria into groups. For example, gram staining. Gram staining differentiates the bacteria into gram positive bacteria, gram negative bacteria. Or acid-fast staining, which is used for staining mycobacterium cells. So it differentiates the bacteria into acid-fast bacteria or non-acid-fast bacteria. So this is one particular category of differential staining, and the other category of differential staining is nothing but it differentiates between the various structural components of a bacterial cell. For example, if you have to differentiate flagella, if you have to differentiate capsule, if you have to differentiate endospore, you have various types of special staining techniques in order to differentiate these particular components of a bacterial cell. So they also come under differential staining. Now we have seen that one form of staining method is positive staining and the next form of staining method is negative staining. What do you mean by negative staining? in case of positive staining, we have observed that bacteria are taking up the stain and they are mainly observed as colored organisms under a bright background, or it is mainly white background. But here, in case of negative staining, the cells remain colorless, that is, they are uncolored, and the background takes up the color, and then a contrast is developed for better visualization of the organisms. So in case of negative staining, we are not staining the bacterial cells, but rather the background is stained and there is a contrast developed between the microorganism and between the background, so that the organisms can be observed clearly. So this form of negative staining uses acidic dyes, because acidic dyes, when the bacterial surface has a negative charge on it, okay, generally the bacterial cell surfaces have a negative charge on it. So when they have a negative charge on it, so they generally attract the positive charge dyes, that is mainly your basic dyes. So when it is an acidic dye which has a negative charge, it is easily repelled. So acidic dyes like Indian ink and nigrosin are used in negative staining methods. So the first step in always your staining is smear preparation. So smear preparation is nothing but it is even distribution of bacterial cells on a slide for the purpose of viewing them under the microscope. So you have to generally prepare an even thin film distribution of bacterial cells on a slide. So how do we do the smear preparation? So you are going to take a clean and a dry microscopic slide, and after you take a clean and dry microscopic slide, you are going to mark an area beneath the surface of the slide. Okay, so that is on the opposite side where you are going to prepare the smear. And after marking the area, so what we are supposed to do is a sample of the culture is placed in the marked area, and then using an inoculating loop, the sample is evenly spread throughout the marked area. So this sample is allowed to air dry, and then this allowed to air dry, and this process is mainly called as your smear preparation. So after the smear is prepared, that is an even film of the bacterial cells is mainly prepared. So you can see here the smear can be prepared from a liquid media as well as it can be prepared from a solid media. So there is a marked area on the slide, and then with the help of the inoculating loop, the liquid media, that is the broth culture, is directly placed in the center of the marked area. But when it is mainly in case of the solid media, so what we are supposed to do is we are supposed to dilute the bacterial culture with a drop of water, and then the smear is evenly spread throughout the surface of the marked area, and then allowed to air dry. So this process is mainly necessary, or the first step in case of your smear preparation. After the smear is prepared, the next important point is mainly nothing but we generally have to fix the smear onto the slide, otherwise it gets washed away when we are washing it with water. So the smear is fixed with the help of a procedure called as heat fixation. So what are we going to do here? We are going to pass this air dried smear through the flame of Bunsen burner 2 to 3 times. So what we do here is we are trying to fix the bacterial cells onto the slide by a process called as heat fixation. So we have learned the first few steps in case of staining, where the first few steps in staining is preparation of smear and then heat fixation of smear. Then we are going to learn about the different methods of staining. So what we observe here is mainly a positive staining, which is simple staining. So what is the purpose of positive staining, simple staining? Simple staining helps you in identifying the morphology of the organism, that is the size, the shape and arrangement of the organism. And when we talk about this simple staining, why is it called a simple stain? Because only a single stain is used which will impart color to the bacterial cells. So when we talk about this simple staining, so simple staining can be positive staining and it can be negative staining. So what we basically see here is the procedure is very simple. We take a slide, clean dry slide. We make a thin smear of the bacterial culture on the slide. Then heat fixation of the smear is done, and then we are going to take it to the staining tray and add the stain onto the smear. Okay, so the stain can be any form of simple stain which can be used, and then we are allowing the stain to retain for 1 to 3 minutes. And after that we are washing the stain, that is you are washing away the stain with water and air dry the smear. So after air drying the smear, you are going to add a drop of oil and you are going to observe the smear under oil immersion objective. This is mainly the procedure for your simple staining. So what we observe here, spread the culture in a thin film, dry in air, then heat fixation, then flood the slide with the stain, then wash the stain and observe it under oil immersion for observing the specimen. So positive staining, what does it do? It mainly imparts a color to the bacterial cells. So you can see here, so on the left hand side, the bacterial cells generally are of a particular shape. Right hand side, the bacteria are of a specific shape. The left hand side picture, the bacterial cells are mainly round shaped bacterial cells. The right hand side, they are generally rod shaped, called as bacilli. Now we have the second important form of simple staining called as negative staining. Negative staining also is mainly used to study the cell morphology, but it is mainly employed where the bacteria are difficult to stain. So for example, we have spirochetes which are very slender and they cannot be visualized by simple staining method. So such type of bacteria which cannot be easily stained by simple staining are generally observed by negative staining. So when we talk about negative staining, as I told you, the background attains the stain, not the bacteria. The bacteria will be colorless and these colorless bacteria can be observed clearly under a colored background. So here the dye what we use is an acidic dye such as Indian ink or nigrosin. This acidic dye will have a negative charge, so it will not penetrate through the cell because of the negative charge. So the unstained cells can be differentiated against the dark background. And in negative staining, there is no heat fixation. Okay, so it is generally not, there is no process of heat fixation in case of negative staining. So what we observe here, this is a clear image of negative staining where we see that through Indian ink or nigrosin, the background is taking up the stain and the bacteria are colorless. So these are the two important forms of your simple staining. Now after simple staining, we have the next important form of staining called as differential staining and undifferential staining. One of the most important example what we always study is called as Gram staining. So it is most widely used staining technique in microbiology to differentiate the bacteria into gram positive and gram negative bacteria. So this particular staining technique was mainly given by a Danish physician, that is Hans Christian Gram, in the year 1883, and this staining procedure was published in the year 1884. It was mainly given by Hans Christian Gram and that is why it is named after him, called as Gram staining technique. So what are the reagents we use in Gram staining technique? So we have 4 reagents what we use in Gram staining technique. The first one, which is crystal violet, also called as primary stain. The second one is mainly Lugol's iodine or Gram's iodine, where it is called as mordant. The third one, absolute alcohol, or it is acetone alcohol mixture, which is called as decolorizer. And the fourth one, safranin, which is mainly called as the counter stain. Primary stain crystal violet, then the second one mordant is iodine, acetone alcohol mixture or absolute alcohol decolorizer, and the last one counter stain safranin. These are the four reagents which are used in the same sequential order in the gram staining procedure. So if we have to mainly study about the basic principle involved in gram staining and how they differentiate into gram positive and gram negative. We have to know about the composition of the cell wall of bacteria. This differentiation of bacteria into gram positive and gram negative is because of mainly the composition of the bacterial cell wall. Basically, in case of gram positive bacteria, what we observe here is the cell wall is completely made up of a thick layer called as peptidoglycan, and this peptidoglycan is mainly consisting of NAM and NAG. Okay, and these peptidoglycan NAM and NAG, basically they have tetrapeptide side chains, and they have cross linking between the side chains to make it a composed layer in case of your gram positive bacteria. So we can say that gram positive bacteria mainly contains peptidoglycan with some amount of teichoic acid which is attached to it, and this is the basic composition of your gram positive bacteria. Now if we go into the details of the cell wall of gram negative bacteria is mainly called as a multi-layered cell wall, where it mainly consists of a complete membrane called as outer membrane. So we have mainly peptidoglycan layer, and above the peptidoglycan layer there is an outer membrane, and this outer membrane is made up of lipid and polysaccharides. So it is mainly given the name called as lipopolysaccharide layer. So as we see here, you have peptidoglycan cell wall, and above the peptidoglycan cell wall you have an outer membrane, and the space which is present between the outer membrane and the inner membrane is mainly called as your periplasmic space. So basically this outer membrane is made up of lipids and polysaccharides. So that means your gram negative cell wall apart from peptidoglycan also has lipid polysaccharide layer. So when we see the composition of gram positive bacteria basically, if you see here, gram positive bacteria has a thick layer, and that thick layer is entirely made up of peptidoglycan, whereas gram negative bacteria, if we see on the left hand side, it has mainly a thin layer of peptidoglycan, and apart from this thin layer of peptidoglycan, it has an other outer layer which is mainly called as outer membrane, made up of lipopolysaccharides. So this is the basic differentiating feature or the principle which is involved in gram staining. So what mainly happens is, basically this particular composition differentiation will allow the gram positive bacteria to stain always purple in color. They retain the purple color and they are mainly called as gram positive. And gram negative bacteria, they basically, what happens is, they basically get the they lose the primary color and they are stained by the counter stain and they appear pink in color. So I will just mainly give you the principle involved here. So what mainly happens here is, so what we observe in case of gram positive and gram negative differentiation here is, the first step is we are mainly going to use a primary stain called as crystal violet. So crystal violet is mainly taken up by all the cells, okay, and the cells stain purple in color. Then the second step, we add a mordant which is mainly called as iodine. What is a mordant? A mordant is a chemical compound which fixes your primary stain to the cells. So here iodine is used as a mordant, and since iodine can fix your crystal violet, then crystal violet iodine complex is formed, and they generally, this crystal violet iodine complex, it will fix the stain into your cells and the cells remain purple in color after the second step. Now the third step is mainly using a decolorizer, which is an alcohol. So this step is a differentiating step where it differentiates between gram-positive and gram-negative bacteria. So what is the principle involved in this particular step? So what we observe here is when mainly a decolorizer is used, that is an alcohol is used. So if gram-positive bacteria are present, they have only peptoglycan layer, and the peptoglycan layer completely becomes dehydrated. So dehydrated, it becomes compact, and the crystal violet iodine complex, whichever is formed in the previous steps, the crystal iodine complex cannot be removed, or it cannot be leaked from the gram positive cells, because the cell wall becomes completely dehydrated due to the alcohol treatment. So they will retain the same purple color. Gram positive cells will retain the same purple color and they appear purple. Now when you have gram negative bacteria in this step, so what mainly happens, alcohol will dissolve the lipids. So it can dissolve the lipids. So the lipid, because your gram negative cells have a lipid layer, that is a lipopolysaccharide layer, all the lipids will get dissolved, and there are pores which are created in the cell wall, and these pores will allow the crystal violet iodine complex to leak out of the cell wall. So it mainly leaks out and the cells will remain colorless. So since the pores become the spores are formed due to dissolve of the lipids, the crystal violet iodine complex which was formed in the earlier step, it will completely get leaked out, and the state is nothing but the gram positive cells will remain purple and the gram negative cells will become colorless upon the third step, which is called as decolorization step. Now when the counter stain is added called as safranin, since the positive cells are already saturated with crystal violet iodine complex, it will not take up any more counter stain, and the gram positive bacteria will be already purple in color, whereas the gram negative bacteria, since they became decolorized in the third step, so they will take up the counter stain called as safranin and they appear pink or red in color. So this is the basic principle which is involved in your gram staining technique. So what we see here, it consists of a primary stain, mordant, decolorizer and counter stain. So it consists of 4 steps. As we see here, the primary staining, it is mainly, that is crystal violet is added and it is retained for 60 seconds, that is 1 minute. Then we wash off crystal violet after 1 minute, then add mordant. Mordant also is retained for about a minute, and then we wash out the mordant, and then we start decolorizing the decolorizing step, and then after decolorization is complete, then we add the counter stain safranin and again retain it for 30 to 60 seconds. Then using a filter paper, we blot dry the slide, and then after blot drying the slide, the drop of oil is added and it is observed under oil immersion lens. So bacteria that manage to keep the original purple color dye, they are mainly called as gram positive bacteria. Bacteria which lose the purple color and take up the red color, they are generally called as gram negative bacteria. So this is how gram staining technique mainly differentiates gram positive and gram negative bacteria. What is the application of gram staining? So it is mainly, gram staining mainly is used for identifying the morphology and types of bacteria in clinical specimens. That is, along with the size, the shape and arrangement, the bacteria also are differentiated into gram positive and gram negative bacteria. So this is the widest application of your gram staining. Thank you. With this, I understand that you have a clear picture of the specimen observation techniques in light microscopy. Thank you once again.